Hey, good afternoon, everyone. I'm Nelson Lau. I'm the director of the Genome Science Institute over on the medical campus and an associate professor in the Department of Biochemistry and Cell Biology. These are my collaborators, Dr. Conizo, Dr. Myers, and Dr. Labradorf, who was one of the speakers but couldn't make it. So I was told I get his extra time. Yes? No, just kidding. I'll try to stay on time. But our study is in a preprint on PubMed here, and my titles is extra long because I'm going to talk about genes and molecules, something very different from all the other talks. Um, I study the expression of selfish genetic elements, which I've represented over here as the transposable elements or transposons over here. Most of your genome, human genome, is made up of transposable elements. Nearly 45% of all the DNA in our genome are these transposable elements. They are selfish. They want to pull against our functional genome over here. But our genome also encodes a defense mechanism called the Peewee proteins and piRNAs that I've represented here as Captain America, trying to push back on these selfish elements. And we've been wondering, could these selfish elements be a root cause behind neurodegenerative disorders like Huntington disease and Parkinson's disease? And we had this really great collaboration with Rick Myers' group, who had generated a lot of matched mRNA and small RNA sequencing libraries from a variety of patients suffering from Huntington's disease and Parkinson's disease over here. And we decided to compare this to normal aging brains RNAs as well. So in this study, it's a lot of comparative analysis and sequencing of transposable elements. And these are coverage plots of the messenger RNAs and the small RNAs coming from transposable elements. This is the pattern from the line L1 transposon. That's the most active transposon in the human genome. Roughly one new L1 is formed every time you are born. So your parents have helped you gain one new line 1 element each time. And they are expressed in the human brain. They make a lot of mRNAs that are then processed into small RNAs with this kind of pattern. And so we'd like to know where these are expressed. In the brainspan atlas, we discovered that most of these transposons are most highly expressed in the neocortex over here, so this portion of the brain of higher thought. And then in collaboration with Rick Myers' lab, we compared data sets from Huntington's and Parkinson's disease, which have very different etiologies. Huntington's is inherited, Parkinson's is sporadic. And we compared these patterns of processing of transposable element mRNAs to small RNAs between normal aging, Huntington's disease, and Parkinson's disease. Just pay attention to these color bars. You can see that Huntington's disease pattern looks like normal aging, but Parkinson's disease patterns is completely disrupted. So in the end, with the summary, what we think is happening is that in normal aging and in Huntington's disease, you have this normal increase of transposon mRNAs that increases while we age, and there's this natural processing of the mRNAs into small RNAs. But Parkinson's disease completely disrupts this pattern. And we'd like to see if this is also the same type of disruption that we might see in Alzheimer's disease to give us some clue as to what might be the molecular cause of Parkinson's disease and other neurodegenerative disorders. Thank you.